substitutions in bacterial genomes.
All right, so hi everybody. My name's Daniella from McMaster University in Canada, and I'm going to talk about my research in spatial patterns of substitutions in bacterial genomes. Now, bacterial genomes are made up of a mosaic of information. This information can come from within the same species or even between species through a process called horizontal gene transfer. Bacteria can further subdivide their genomes into multiple replicons or chromosome-like structures that allow for meeting specific needs of the bacteria, such as adapting to a local environment or metabolic needs. Now, because information is coming from all over the place, different positions in bacterial genomes can have varying molecular traits. A number of studies have looked at this when moving from the origin of replication to the terminus, and have found that mutation rate tends to increase with increasing distance from the origin. So there's higher mutation rate near the terminus compared to the origin, whereas gene essentiality, gene expression, and gene conservation all tend to decrease. Now, a few studies looking at mutation rate in particular didn't find any sort of correlation between distance from the origin and mutation rate, but they did find that it varies. So depending on the genomic location of a genetic element, it can vary their molecular traits, such as expression or mutation rates. Now, the issue with these studies is that they were done using a limited number of genomes, sometimes just one bacterial genome per species. Now, with the increase in sequencing data, we now have multiple genomes available, so we can increase this information and look at these trends with more genomes. However, when you increase the number of genomes or bacteria that you look at in a study, a number of complications arise. So we have varying genomic structures in bacteria. They can have their genomes organized into circular, linear, or multi-repliconic. And when you increase the number of bacteria that you look at, you increase the combinations of these genomic structures that you can have, which can complicate things. We also have an increase in the number of rearrangements and inversions that happen often in bacteria. These diagrams just depict whole genome alignments of 5 different strains or multiple strains of Rhizobium mellilodi. Each line represents a different taxa, and the colored blocks and corresponding colored lines represent different locally collinear blocks. So on the left-hand side, we have 5 strains of Rhizobium. You can see that there's a few rearrangements and inversions, whereas on the right-hand side, when we increase the number of genomes to 12, we see that there's many more rearrangements and inversions. So these are happening frequently in bacteria, and when you increase the number of genomes, they happen often, so it's important to account for them. Now, my research focuses on what spatial trends in substitutions are seen in bacterial genomes, and I take a novel approach at this by accounting for rearrangements and inversions through the ancestral reconstruction of both nucleotides and genomic positions, as well as through a genome alignment program that takes into account these rearrangements and inversions. Now, the expectation, based on previous studies, is that number of substitutions should increase with increasing distance relative to the origin of replication. So my analysis can be broken up into 2 main components, the substitution analysis and then a supplemental spatial gene expression analysis. For the substitution analysis, downloaded 25 complete bacterial genomes from E. coli, Bacillus subtilis, Streptomyces, and Rhizobium mellilodi. These bacteria represent a variety of different living environments as well as genomic structures. So E. coli and Bacillus are both circular genomes in nature, Streptomyces is linear, and Rhizobium is multi-repliconic. Then aligned each of the genomes using progressive move, which accounts for local and large-scale rearrangements, by splitting up the alignment into locally collinear blocks. These blocks were then realigned with MAFT to get a better local alignment. Then we created phylogenetic trees using phylip to assist in the ancestral reconstruction of both nucleotides and genomic positions, and this was the main component that accounted for those rearrangements and inversions that are happening so frequently. The genomic positions were reconstructed using the CodeML program in PAML as well as a Perl wrapper, and this just allowed us to determine the genomic positions of all the nucleotides in the alignment in the ancestors. And then we also reconstructed the nucleotides using the BaseML program in PAML that allows for multiple substitutions. So this overall gives us a picture of what substitutions were happening in the ancestors as well as their locations in the genome to see how things are being shuffled around. Now, all bacteria replicate bidirectionally, so begin replication at the origin and continue in both directions around the replicon till it meets the terminus. So this bidirectionality of replication was accounted for by ensuring that all genomic positions were relative to the origin. We then removed any outliers and gaps and performed a simple logistic regression to determine if the number of substitutions were increasing or decreasing when moving away from the origin of replication. Then for the gene expression analysis, the same bacteria were used. We downloaded raw control expression data from the gene expression on Nimbus, normalized this using a TMM normalization method, calculated a singular genomic position, so each gene had one position, which was denoted as the midpoint of the gene. Again, this bidirectionality of replication was accounted for by ensuring that all genomic positions were relative to the origin, and then performed a linear regression to see if gene expression was increasing or decreasing. So this table summarizes the results from the substitution analysis. You can see that for majority of the replicons, the results are negative, indicating that the number of substitutions decreases when moving away from the origin, which is opposite and not expected based on the previous studies done with mutations. And then for the gene expression analysis, again, we see that majority of the bacteria have a negative trend. So again, the gene expression decreases when moving away from the origin of replication, which matches what previous studies have found. So to summarize, for both the number of substitutions and gene expression in this study, for majority of the replicons, we see that both of those trends tend to decrease when moving away from the origin of replication. There were a few replicons that were exceptions to this that are highlighted here in pink. Now, some reasons why we might be seeing this surprising decreasing substitution trend is that transposon insertion events tend to peak at the origin and are at a minimum at the terminus. So this means that new genetic information is being brought into the origin, which could bring with it different genomic signatures such as asymmetry in nucleotide composition, differences in GC content, or even differences in substitutions. So this could cause a spike in the number of substitutions near the origin relative to the terminus, potentially describing why we're seeing this trend. As well, potential genomic and pathogenicity islands are also found near the origin in a variety of bacteria, and this again can bring new genetic information that has different genomic signatures, which again could cause this potential spike in substitutions. And then additionally, bacteria undergo multiple rounds of replication during one replication cycle, so there's the potential to have multiple replication forks in one cycle. These could be concentrated near the origin, and we know that these replication forks are hotspots for rearrangements and translocations, which again could bring new genetic information with different genomic signatures, potentially causing this decreasing substitution trend. So to kind of summarize, previous studies found that when moving from the origin of replication to the terminus, mutation rate tends to increase and gene expression tends to decrease. And then I have confirmed the results with gene expression, but found the opposite trend with respect to substitutions. And with that, I would just like to thank a couple labs at McMaster, as well as NSERC for funding, and thank you for listening.
Yeah, we can have one question.
Maybe the next speaker can already come.
So, very interesting talk. Interestingly enough, actually in mitochondria, you get considerably more substitution next to their origin of, to their own genome. Origin of replication is the highly variable regions if you go from the ORI site. I was wondering if you can comment about that. Have you thought about that? And have you looked at other bacteria?
We haven't looked at mitochondria or any other of the bacteria, but that would be interesting to, because obviously I'm seeing similar trends, so it would be interesting to look at the mitochondria and see kind of what.
Yes, because they're kind of the opposite in that respect.
Yeah.
So it'd be interesting to see why that is.
It's so
Yeah, okay, thanks.
Yeah. Thank you.
Okay, thank you for a nice talk. And